This is RNA splicing. In RNA splicing, our strand of pre-RNA consists of 2 exons and an intron with a 5' cap. In RNA splicing, proteins combine with sRNA to create snRNPs, also known as small nuclear ribonucleoproteins. The snRNPs combine with other proteins to create spliceosomes. The spliceosome acts as a riboenzyme. A riboenzyme are RNA molecules that can act as enzymes and splice out RNA. So, the spliceosome will find the right intron, and using base pairing, it will splice out the intron. The spliced out intron will later be degraded. As for the remaining 2 exons, they will be spliced together by proteins with a 5' cap, in addition with a poly A tail. This structure of the finalized mRNA will allow it to be stable as well as leave the nucleus through the nuclear pores. So this is an inducible operon in negative gene regulation. The first step in this inducible operon is that LAC-I, which is a regulatory gene, codes for mRNA, which then translates to an active repressor. The active repressor is a protein that will bind to the operator. This is an active repressor. When lactase is present, L-lactose will enter the cell, which is an inducer, and L-lactose will bind to the active repressor and change the shape of the repressor. These two combined will now become an inactive repressor. So now that the repressor is out of the way and the operon there's nothing blocking the operon from transcribing, RNA polymerase will then go through and translate all of these genes. RNA polymerase will transcribe these genes. LAC-Z will translate into beta-galactosidase. LAC-Y will translate into permease, and LAC-A will translate into transacetylase. Beta-galactosidase causes lactase to change into glucose and galactase. Meanwhile, permease is a surface protein that allows lactase to enter, and these inducers allow lactase to enter the cell, which then that's negative feedback because it causes the active repressor to become inactive. So this is a type of negative gene regulation. This is a repressible operon. This specific repressible operon is the TRP operon, also known as the tryptophan operon. So what happens is that this operon starts off with RNA going through the operon and translating all these TRP genes into proteins E, D, C, B, and A. Now all these proteins will eventually contribute to the process of making tryptophan. Tryptophan is also a co-repressor. Now earlier in this strand of DNA, there's a regulatory gene that codes for an inactive repressor. So what happens is that tryptophan, which is the co-repressor, binds to that inactive repressor. And as a result, it will change the shape of the inactive repressor, and this will become an active repressor. Now, when this has occurred, RNA polymerase will not be able to translate any more genes in the TRP operon, and all of this will stop occurring. So this is negative gene regulation in that the product of this operon, which is tryptophan, is the cause of its own stoppage. So we're going to talk about enhancers. Enhancers are just distal control elements, or control elements that are far away from the promoter. Control elements may just be specific nucleotide sequences. So, and what enhancers will do, they will bind to protein activators. And then DNA bending protein will bend the DNA over so that the enhancers and the activators are closer to the promoter. What happens next is that mediator proteins will bind to these activators. Followed by that, you're going to have adrenal transcription factors bind to these as well. And this causes RNA polymerase to bind to the promoter region and start transcription. So this is called an active transcription initiation complex. What this does is that it speeds up or it amplifies RNA synthesis